All right, so lastly in Chapter 19 that we're going to talk about are healthcare-associated infections, or HAIs. These are infections that are acquired while in a medical setting, so like a hospital, a nursing home. And healthcare or nosocomial infections are often spread by either a patient's own normal microbiota or by other sick patients, because when you're at a hospital, typically you are already immunocompromised. These are some of the most common microbes that cause healthcare associated infections. One of them I always like to show is Pseudomonas. These actually grow in environments that most other microbes couldn't. So they can grow in disinfectants as well as in some of the soaps that are used to clean items. And because of this, these are one of the most common organisms to cause things like urinary tract infections. Hold on one second, I have to fix the slide. So what spreads HAIs? Typically, it's either the patient's own body microbes or it's other patients. So remember, in hospitals or in other healthcare environments, you're going to have a lot of microbes that are, or you're going to have a lot of immunocompromised patients. And there will also likely be more microorganisms because a lot of these patients are sick with different microbial diseases. Another thing that can spread diseases are the healthcare workers. Healthcare workers themselves can come in with disease. So if they have a cold or if they have some type of an infection, they can be carrying those diseases to their patients. And so I found this, I thought this was cute. Here we have the hospital staff, their support staff. Here's MRSA, here's Streptococcus, and then here's another one that I thought was cute. Hey kid, want to be a superbug? Stick some of this into your genome and even penicillin won't be able to harm you. So here we have conjugation going on between two different microbes. It was on the shortcut through the hospital kitchen that Albert was first approached by a member of the antibiotic resistance. Kind of cute. And nowadays, because HAIs are so common, hospitals and other medical environments have infection control committees that just monitor HAIs and put policies into place to help decrease the likelihood that HAIs occur. It's only been 4 minutes. I'm not going to stop this lecture and go into and upload it. So instead, I'm going to go right into Chapter 20. Chapter 20 is all about antibiotics and where antibiotics came from, and then how they are used to treat different infections. So what are chemotherapeutic agents? These are agents that are used to treat disease. They're chemicals used to treat diseases. They're either going to kill outright different microbes, or they inhibit the growth of microbes, and then the host's immune system will actually kill off the pathogen. Most chemotherapeutic agents are antibiotics, and antibiotics, that term means anti-life, but typically when you think of antibiotics, we're treating bacterial infections. Um, and antibiotics are produced by one microbe and is used to kill another microbe. So what we're going to focus on is where these antimicrobials came from and how they work to treat infections. One of the first antimicrobial drugs was called Solverson. Solverson was discovered by Paul Ehrlich. This is around 1910, and it was used as a treatment against syphilis. So it treated the primary stages only. Syphilis has three basic stages: primary, secondary, and tertiary. So primary stages were the first stage, when you saw there were like cankers that would be found on the body. Sometimes, well, there you can see a canker on this tongue. You might find a canker in the genital regions or other regions of the body. That was the first sign that you had syphilis. After this, the cankers would go away, and a few weeks later, you may have a secondary rash, but sometimes you wouldn't even see that secondary rash. After this, the syphilis bacteria would actually stay in your body, in the cells, and you would have a period of what we would call like a remission, where the bacteria would live in our cells, but there would be no sign of infection. And this could last for years and years. Tertiary stage syphilis caused severe brain damage and death of the individual. So Solverson was able to treat syphilis if you caught it at that first primary stage. Around 1930, Gerhard Dogmack found sulfa drugs and learned that sulfa drugs could treat streptococcal and staphylococcal infections. The first drug the drug that he actually found was Prontosil, and when he found it, he found that this drug could treat streptococcal meningitis, and it was very effective at killing off these bacteria. Then Alexander Fleming found another drug or chemical. This is the first actual antibiotic, and he discovered it by accident when he left some of his petri plates by the sink. These plates had Staphylococcus on the plate, and what he noticed was there was this ring of inhibition where there was no staph growing around this fungus. And so he then identified the fungus as, as penicillium. The penicillium had bactericidal agents, and he found out that, hey, this penicillin is actually very effective on a wide variety of mainly gram-positive microbes. And the reason for this is because, and we know this now, penicillin inhibits peptidoglycan synthesis. The problem was, though, Fleming couldn't purify it. And because of this, you couldn't use it to treat actual infections. So he abandoned his research, and later on, Howard Florey and Ernest Chain came along, and they asked if they could take a stab at purification of the penicillin. And so they did at the end of the 1930s. And they were able to purify it, and so then in 1941 they tested it on their first human subject. This patient was ill. He had been working in a flower garden and had just been scratched. That's it, by a thorn. And he became very deathly ill, ended up in the hospital. They started using the penicillin on him, and he got better. But there wasn't enough penicillin to treat the infection and cure him. And so he got better. They ran out of penicillin. He got worse and he died. So this was super, super bad. This is also right around the time of World War II. And they wanted to have a lot of this penicillin to, test out and then to be able to use if it was really effective. So they got a bunch of factories involved in mass producing the penicillin. And they produced enough penicillin, they were able to then retest it on a new patient. The patient fully recovered, and then they were able to use it to treat infections during World War II. So this was like the treatment, and it saved soldiers all around the world. So this was like huge. In 1945, Fleming, Florey, and Chain all received the Nobel Prize for their discovery and purification of this first antibiotic. So now we know, hey, these microbes produce antibiotics. Selman Waxman then found another antibiotic, streptomycin, from Streptomyces griseus, and this was the first antibiotic used to treat tuberculosis. And it was also able to treat some gram-negative bacterial infections. So now we have these antibiotics, and people were like, whoa, antibiotics are the thing. We don't have to worry about bacterial infections anymore. Anytime you go to the hospital or to the doctor, they'll just give you an antibiotic and you'll be better. And so, it went on like this for a long time, actually. The main things we were worried about was how toxic is the antimicrobial drug that we're using, because you have to look at, okay, if the infection is dangerous, we have to treat it, but if the chemical we're using to treat the infection is worse, we're not going to use it. And that should make sense to everybody, hopefully. If it doesn't, then ask, of course. If antibiotics cause more harm than the microbes, then you're not going to use the antibiotic. That's basically what we're looking at, okay? In general, antibiotics are going to cause more problems to microorganisms than to humans. And this is because they're attacking biochemical processes that are common to bacteria and not humans. We have to look at the toxicity of a drug and determine, if it is beneficial for a patient to use it. So if an antibiotic has a very high toxicity, then we're probably not going to use it. The lower the toxic levels to patients that they are, the better it is for the patient to use. So any drug that has a high therapeutic index typically has a very low toxicity to patients. We also are looking at how well the antimicrobial will work against microbes. So some antimicrobials are going to work against a wide variety of microbes, while others are going to be very specialized. Narrow spectrum antibiotics are going to be used on a small range of microbes, maybe only gram positives or only gram negatives, or maybe they're even more specific to they only treat mycobacterium. So these narrow spectrum drugs may not be as beneficial amongst all microbes, but they're going to really work well to kill off specific microbes. Broad spectrum antibiotics work against a lot of microbes, but they might not be as effective as a narrow spectrum would be against its own microbes. So I always look at myself and one of my colleagues when I think of broad and narrow spectrum antibiotics. My colleague can teach almost any biology class out there. So she would be considered a broad spectrum biologist. She can teach general biology really effectively. She teaches the environmental science classes. She teaches microbiology. She teaches A&P fundamentals. I'm trying to think of the other classes. Zoology. So she teaches a little of everything. I am more narrow spectrum. My main classes that I teach are anatomy and physiology and microbiology. Beyond that, I can teach other classes, but I'm probably not as effective as she is at teaching other classes. But when I teach my classes, I'm really good at what I teach. I can do micro and anatomy really effectively. And my students tend to understand the material relatively well. So I might not be great at teaching zoology or environmental science like she is, but what I do teach, I teach really, really well. Broad spectrum, like I said, it can teach a lot of things or can work on a lot of different organisms, but they might not be as effective as that narrow spectrum would be on their one specific thing that they kill. A disadvantage of broad spectrum antibiotics is that because they're broad spectrum, they might mess with your body's normal flora as well. So what are some of the adverse effects that you can have then when taking antibiotics or antimicrobial drugs? Well, I just talked about suppression of the normal flora. If you're taking a broad spectrum antibiotic, you're going to suppress your normal microbes. And that can cause other problems because when you kill off your normal flora, then other microbes that are being kept in check by your normal flora can become more common. And that's where like yeast infections become more likely to occur. Allergic reactions. Some individuals are actually allergic to certain antibiotics. The most common allergic reaction is against penicillin and amoxicillin because penicillins actually can act as, what is the term? They can basically bind to a host's molecules and become antigenic. So that they're considered a hapten. And a hapten alone is not going to cause any problems. But for whatever reason, in certain individuals, the haptens bind, the penicillin binds to a host molecule and becomes an antigen or acts like an antigen, and our immune system responds. Some chemicals can have toxic effects. They can cause our body to not be able to absorb the proper medicine or the proper vitamins, and then we won't be able to produce normal proteins. And the example I use here is aplastic anemia. If we take certain antimicrobials and they kill off our normal flora, then some of our normal flora, i.e. E. coli in our gut, helps us to produce and absorb or to absorb the vitamin B and vitamin K that we're supposed to have. Vitamin K is needed in producing blood cells. And so without, oh, and so is vitamin B. But without these vitamins, then we wouldn't be able to produce red blood cells properly. And then suppression of normal flora, I already talked about this, allows other organisms to become more common. The last is antimicrobial resistance. So antimicrobial resistance occurs when microbes become adapted to your, or not adapted, I'm sorry. Microbes adapt to the environment and can grow in the presence of antibiotics. So I'm going to stop here and then I'll upload this video and I'll get the next one started.